this opportunity to welcome, thank you, Melissa, to welcome everybody on the second episode of the Engagement Forum of in 2022. This is a forum where we are really looking into a range of concepts that relate to research and engagement in communities. So it is really meant for all of those who are involved in this work. And in the last one in February, I think in January, I'm already in March, we had a discussion about how we can be ready for what it is that we have learned in the past 2 years of working in HIV prevention research, but within the period of COVID, which brought a lot of interesting changes and dynamics, and how it is that we can adapt and continue to really do this work whilst the focus on COVID as well is continuing. And we really had a range of panelists really speaking to us about that. And today's topic is a topic that we think most of us are asking, especially if you have been involved in HIV prevention research work, and in fact any other HIV-related research and TB, where we have come up with a number of messages, especially when working with communities and advocacy groups, where we have really had to present research literacy about how research happens, why is research important, and one of the aspects as part of that community engagement and outreach work is to really explain a number of concepts. And one of those concepts is the concept we're talking about today, which is a concept we're learning that from communicating how trials are designed. We have often spoken about a trial being a placebo-controlled trial, but today we're learning that there are developments with regards to new trial designs, and some of those developments include the fact that because of the success in the HIV prevention field, where we now have PrEP, and there are a number of issues that relate to ethics and how trials are designed, where a placebo cannot no more be used in certain circumstances. And we're really going to be talking about that in really trying to understand these new trial designs in HIV prevention research. So as you can see, we are today joined by Professor Ayesha Kasani, who is a specialist scientist and epidemiologist at CAPRISA. And Professor comes with a PhD in medical microbiology and has a master's training in epidemiology from the Columbia University in New York. She has a lot of experience in the field of research and in teaching, professional and leadership experience, and takes a leading role in the field of HIV surveillance and prevention studies. Her area specifically of expertise is on understanding the evolving HIV epidemic, and that's really why we have Prof today. And she's done that work in South Africa, especially on HIV acquisition in young women. She also was a PI on the CDC PEPFAR-funded HIV incidence provincial surveillance system, and she set up a HIV surveillance platform to evaluate the impact of the contemporaneous programmatic scale-up of prevention and treatment efforts in a real-world setting. And there's really a lot. I can go on and on and on in really presenting Prof, and we are very excited to have her today really take us through a number of these concepts that relate to new trial designs in HIV prevention trials. Again, this is an engagement forum that's brought in partnership between Advocates for Prevention of HIV in Africa and the Desmond Tutu Health Foundation, with partners that include the VETS RHI, SATAC, the Vaccine Advocacy Resource Group, and AVEC. Professor, we will hand over to you just now, but as usual, please, this is a conversation. We're going to give some time to Prof to really take us slowly through all of these concepts and give us a background actually of how we got to be here today. And then in the meantime, please feel free to introduce yourself using the chat box, and please keep yourselves muted, and then continue as we listen to Prof. All of the questions that you have, let us put them on the chat box. And welcome everybody. And without further ado, we'll hand over to you, Prof.
Thank you very much, Ntando, and it's a pleasure to be talking at this forum, and I hope to take you through some of the information that I have, and at the same time, please feel free to keep your questions, and we will try and answer them or respond to them as much as I possibly can over a period of time as well. So let me just get into this presentation. And so, as Ntando already explained to you who I am and where I come from, and my passion has been in HIV prevention research, but today I'm going to talk specifically on, if not placebo-controlled, what understanding new trial designs in HIV prevention research. So, overview of this presentation is basically going to involve a bit of background, some information on the HIV epidemic, with some definitions that we've had, HIV prevention interventions, how we determine the effectiveness of potential interventions, challenges in assessing products, measurement of HIV incidence, some methodology, and then going through some examples as well. I just felt it was important to somewhat recap on our HIV prevention research, specifically in the testing of interventions. As I think over time, we tend to forget some of the nuances, and then it's just a quick reminder of where we are as well. So I think this is clearly what we all know about, and why we are in the field as well, is that we still, in South Africa, we still have 1 in 5 new infections. And of course, I see we have almost an international forum here. But to remind ourselves as well is that sub-Saharan Africa remains the worst affected, and South Africa has 1 in 5 new infections. And these new infections are spread, they are unevenly distributed as well, in that the province of KwaZulu-Natal has a higher rate of infections compared to some areas like the Western Cape, etc. So the key goal is to prevent new infections. And that has been our guidance from the very beginning as well. Yeah. So just in terms of some of the definitions that we use when talking about HIV as well, I think many of us know it, but just to recap as well at the same time. So when we talk about HIV prevalence, we talk about how much of disease we have at a given point in time. So this is measuring existing HIV positive people. And usually these individuals are identified at screening in our PrEP trials as well. The important thing about prevalence is that we have no indication of when infection was acquired or how long they have been infected, how long they have been living with HIV as well. But HIV prevalence as a measure is an important measure because it has implications for health impact, and it also indicates the need for healthcare as well. The next measurement that we use is HIV incidence. And HIV incidence gives us a measure of how many new infections are acquired in those that have not had the infection. And the incidence rate is the HIV incidence rate is a rate at which new infections are acquired over a specific period of time as well. So this gives us an idea in terms of how we can compare with different countries, etc. It's a measure that allows us to compare as well. It's also a more sensitive measure for assessing growth of the epidemic, and it's a sensitive marker to assess effectiveness of interventions. And this is a key measure that we use in our HIV prevention trials, in any of our prevention trials as well. So over the last several years, in fact, for the last decade or so, or more than a decade or so, we started having interventions, and We started off with, for example, with condom prevention, MMC, etc. But we know that those interventions are generally male dependent. However, with the interest in ARV, with microbicides and ARV-based microbicides and oral and PrEP as well, we've now gone into the field of looking at ARV-based interventions. And so the first study that looked, I mean, over the last several years, we've had a number of studies as well. But the first study, the first successful study that looked at a PrEP intervention was Truvada. So once Truvada became available, it became the standard of care as well. This was followed by dapivirine ring and of course cabotegravir, and there are many more interventions that are actually being assessed at the moment as well. So when we determine the efficacy of randomized controlled trials, we need to determine either the efficacy or the effectiveness of these interventions. And the key method, the methodology that is used to assess these interventions is the randomized controlled trial. The randomized controlled trial is, in terms of the hierarchy of the strength of evidence, this is the highest in terms of the evidence that it provides us. So we have a sample population, and that sample populations, once it enters our trial, they are sort of randomized into group 1 or group 2, and it's usually a random process. It's not dependent on the investigator, neither is it dependent on the participant themselves in terms of any choices as to which intervention, which group they would prefer to be in. So once the groups are randomized, group 1 could go into the new treatment, group 2 could go into the placebo. And it's not by choice, it's just by randomization. Once they are on these interventions, we follow them through and we look for HIV incidence. So these are HIV negative people who have entered our study, they've been randomized to these interventions, and over a period of time, we try and determine whether they become HIV, they acquire HIV or not. And that gives us an indication whether the treatment has any efficacy, whether it is effective or not. So again, just to remind us, it's a gold standard of evidence. It's a prospective study. That means it is going forward in time. It is to test the effect of a product. It's the active product versus the control product. The control can be a placebo, or it can be another effective product as well. So what is the placebo then? The placebo is actually the dummy treatment that looks exactly like the treatment. It is the same in color, shape, or size of the injection, or the pill, etc. And however, the placebo is ineffective in comparison to the intervention itself. Now, the placebo also helps to assess the true effectiveness of the intervention. So we have to compare our intervention to something else, and that's the reason that we use a placebo. In addition, we have this blinding where participants are blinded to whether they receive the intervention or the placebo, and that blinding and the placebo helps us remove the bias in terms of the study itself going forward. So there are several ethical considerations for the use of placebo as well. And placebo can only be used if there is no proven effective treatment for the condition that is under study. So we cannot use a placebo if we have an alternative treatment. It can only be used if we have no proven effective treatment. And therefore, all our initial trials on HIV prevention research has been conducted in the context of using a placebo. Therefore, we need to provide the next best option and test against new interventions as well. So once we have a placebo, we cannot actually use once we have an intervention available, we cannot use a placebo. And of course, then we have to test the new intervention against the existing intervention itself as well. So just very quickly to remind ourselves is that, we don't just get into phase 3 large clinical trials. It goes through a series of smaller trials where the safety of the intervention is determined, whether the efficacy, whether there's any indication of safety and in terms of the dosing strategies that would be required, etc. And then we go into the large effectiveness trials, and then those are actually followed by the phase 4 trials, where if we see our Sisonke study, that is an equivalent of the phase 4 trial going into the field as well. So it's just to remember is that, it's a long procedure. It just doesn't happen overnight. It takes many, many years to get into this phase 3 trial as well. So it's a lot of energy that goes into these studies as well. So when we look at our HIV intervention studies, and these are some of the interventions that have gone through testing, and the one that we have in the red box that I've highlighted here are the interventions that showed us effectiveness in either in men who had sex with men or in HIV serodiscordant couples as well. So the Truvada study, these are all the Truvada studies, and then the daily tenofovir as well. So the Truvada studies were the first set of studies that showed us that Truvada was effective in preventing HIV acquisition. And this was at a rate of around 86%, but it was slightly lower amongst women, but amongst serodiscordant couples. And therefore, at this point in time, we know the effectiveness of Truvada in the MSM population, but less so in the female population. And I'll just get back to that once we go through. So now that we have these Truvada as an intervention, we cannot use a placebo because Truvada is now the standard of care, and going forward, any intervention that is going to be tested has to be tested against Truvada itself as well. So these are some of the PrEP trials, and to look at in the context of what in terms of the clinical trial design. So as I mentioned initially, is that we have an intervention that is being tested, and that we don't have any standard of care at this point in time. So we will test it against a placebo. And so the framework of that particular trial is a superiority trial. So because there's no proven, so this is the trial. Initially, we had no proven effective intervention. However, now that we have Truvada as an intervention, we cannot use the intervention versus a placebo because we now have Truvada, and then therefore the direct observation is of seroconversion has to be in the context of Truvada. It's Truvada and a new intervention as well. So now that we have an intervention that is now available, for example, if we have Truvada, we would be looking at a new product itself as well. So a proven product. So we would have the new intervention and we would have the proven product, for example, like Truvada. So that kind of a trial is called a non-inferiority trial because we know that the intervention, by the time it goes through to a phase 3 trial, we expect it to be highly effective as well. And we know that the effectiveness of Truvada is also around 80-some-odd%. So we have these two interventions, the intervention that is being tested, and we have the proven intervention that has very high levels of effectiveness. And therefore, for this reason, we will need extremely large sample sizes to show that statistically significant difference over the period of time. We need large sample sizes. We need a prolonged period of time that we will be testing these interventions for. So what happens is that because Truvada is now available, it will be unrealistic to obtain the required sample size to observe this difference because of the effect, the highly efficaciousness of these interventions. Again, in these kind of trials, we actually measure it's a direct measure of HIV seroconversion of individuals. So people get into this trial, we follow them up over a period of time, and we look for HIV seroconversion as well. The next method of measurement would be the counterfactual background HIV incidence. So what does counterfactual background HIV incidence mean? So this is what we want to know is that what is the HIV incidence in a similar population? For example, if we're looking at an MSM population, or we're looking at adolescent girls and young women, what would the HIV incidence be in a similar population if we do not include PrEP? Now, we cannot do that because ethically, we're obliged to use PrEP if these individuals are in our trial. But we're looking for HIV incidence in a similar population, but these people are not necessarily in our trial. So that is the current option. And the way we would measure the counterfactual background HIV incidence is using a biomarker method or which is the recency assay. So we're not measuring the direct observation of seroconversion. We're not getting people into the study, following them up over time and looking for seroconversion. These individuals would already be HIV positive, and we're using a biomarker to determine whether the antibodies that they have are strong enough or weak enough. And that's the method that I'm going to show you right now as well. So, sorry, before I actually go into that as well. So just recapping some of the trial designs as well. So in this case, we would have, for example, we now have Truvada. We have Truvada, which was tested in the context of a placebo. And when it was tested, we know that the group in the placebo, the HIV incidence was around this is a hypothetical example that I'm giving you as well, was 6.4%. And in the Truvada arm, it was 2.4%. So this difference is a statistically significant difference, and therefore the reason for Truvada going into policy and practice as well. So now that we have, as we start looking for choices and increasing choices, we have new interventions being available as well. So this would be the new intervention versus Truvada, because we cannot use placebo now. It would be unethical to use placebo. So we compare the new intervention to Truvada. And as we get people with HIV uninfected individuals onto these interventions, we follow them up over time. And this is the incidence rate that we have. We have 2.5% for the one and 2.4% for the other. The difference between these two HIV incidence rates is so low that we cannot say whether these are not statistically significantly different, and therefore we cannot say the one intervention is better than the other intervention. So can you see, but both are excellent, but how do we compare this? So we have to have some kind of an HIV incidence rate for us to be able to compare. So both these interventions can go into the field, into policy and practice, but it has to be compared against a group where we know that because when we are undertaking these interventions, we don't know whether something else from the outside world could have impacted on these incidence rates as well. So we have to compare it to something else. And therefore we go on to the counterfactual HIV incidence rate as well. So this is the background, counterfactual is the background HIV incidence rate in a similar population in the community itself. And I'll go through how we measure that as well. But just to give you an example, is that this is the HIV incidence rate after follow-up of individuals, which is 2.8 and 2.6%, again, is a hypothetical figure. And then we would have the counterfactual HIV incidence of 6.2%. And here we would, so what this would indicate to us is that in our communities, the HIV incidence rate in this given population is still high. And therefore, the incidence rate as a result of the new intervention and Truvada is showing the true effectiveness of these two interventions in comparison to the background intervention that we had. So this is the way we would measure, this is the way we have evolved in terms of measuring HIV incidence based on the original study, based on the potential new studies, and as we go forward, potentially using the counterfactual HIV incidence rate as well. And so just to remind ourselves as well, we get onto these trials and we're following individuals, we're doing all the hard work, but just to remind us how we actually derive the HIV incidence rate following seroconversion. So we would have our study sample and we would screen this population for HIV. All our HIV positive individuals will be excluded. All the HIV negative individuals will be enrolled into our study. They will be randomized to either the intervention or the placebo or to the new intervention or to either the intervention or the placebo, and then we would follow them up in time. As we follow them up in time, we would get, for example, we know individuals may not have used, may not have been adherent, etc. And for whatever reason, some individuals acquire HIV. Whereas some individuals will continue with the study and they will end the study not acquiring HIV. Some individuals will during the early stages of HIV being into the trial may acquire HIV, whereas some individuals, as you follow them through, for some reason or the other, they are lost to follow up as well. So this is the way we would measure HIV incidence in a longitudinal way, especially in our randomized controlled trials. So this type of a study is therefore very time consuming. It is extremely costly, but it is the gold standard of measuring new HIV infections as well. So again, remember in my very second slide, I talked about new HIV infections, incident infections, or seroconversions, and this is the way we would measure new HIV infections as well. What about measurement of using the counterfactual measurement of HIV incidence and measurement of background HIV incidence? And so this is looking at a biomarker method. It's the recency assay for the measurement of HIV incidence. And this measurement of HIV incidence is usually undertaken from cross-sectional studies. And this is just to give you an indication of a design of a cross-sectional study that I have here below. So the test that is being used and the test that is being, there are several tests that are being that are available, but the limiting antigen avidity enzyme immunoassay is being used, which is the lag avidity assay. This assay measures the maturity of the HIV antibody response. And when we measure the maturity of the antibody response, it gives, the test is actually measuring the avidity. It's a concept, the strength of the antibodies. So it's the antibodies of low avidity are then determined to be recently acquired infection. So we would, on these cross-sectional sample, we would have all our HIV positive individuals will be offered, will go through the limiting antigen avidity assay. If the avidity is low, these will be classified as recently infected individuals. If the avidity is high, then they are considered to be past infections. All those with low avidity infections then go through another algorithm to determine whether they are truly new infections or not. This is a little bit complex. I'm not going to go through all the detail in terms of the complexity of the methodology, but basically to say that it's a biomarker method. It's undertaken from cross-sectional studies, and it measures the maturity of the antibody response, and those of low avidity are considered to be recent infections. And therefore, these then, this is then your counterfactual background HIV infections that we are then measuring as well. So it's different from the longitudinal follow-up, where you're looking at one individual that acquires HIV over a period of time. But in this case, the counterfactual HIV incidence is based on individuals who are already HIV positive, but you're measuring the avidity of the HIV, of the antibody response as well. So some considerations about the counterfactual as well. So I think it's important, as we are transitioning from a concept where we conducted randomized controlled trials, following up following up individuals, and here we are looking at individuals who will be part of our study, but we will also be undertaking additional tests on those that test positive. So we need to actually make sure that everybody understands the concept, and therefore all stakeholders must be aware of the trial designs. And this includes, I mean, Kanda will give you some information. It includes just about everybody. It's all our community members, etc. Our participants need to know that as well, and that what we are actually measuring as well. And of course, because the concept has been sort of different in the past, we need to make sure that our regulatory and our ethics committee understand those concepts as we are going forward as well, in terms of what the background information is on HIV incidence and how we look at them as well. And what the methodology will be, we will be using, how we are going to estimate them, and how we are going to be triangulating these data to give us the measurements that we will require. What I'm also, I think I haven't brought this in, but I think much of this information is very much statistics dependent as well. So the statistical criteria in terms of the sample sizes that are required, and how do we evaluate the efficacy, etc., over time as well. So again, all the stakeholders need to be aware about it. It's something that I also recently started reading into it, and have been sort of been aware of it from, in fact, from the last IAS meeting where we participated in this forum, where we were looking at these counterfactual trial designs as well. So now, just moving into some of the phase 3 designs, especially for looking at the newer products for adolescent girls and young women, and of course, trying to obtain choices as well. We know that there are several studies that are, there's several products that are actually getting into the field. We have the lenacapavir study, we have the tenofovir alafenamide study as well. And of course, we have the current Truvada that is existing as well. So the Truvada, we know for MSM and transgender women, it's highly efficacious. For heterosexual men and women, it is also highly efficacious as well. Whereas the tenofovir, we know about the tenofovir, but we less so amongst heterosexual men and women, and therefore it's continuing with these trials as well. The other information that we sort of focus on as well is that these two are based on daily dosing regimens, but obviously we're looking for products that are long-acting, and so lenacapavir is also the long-acting product which needs to be provided every 6 monthly. It's currently going under testing, and we need to make sure that we are currently understanding the concepts of these trials as well. So just to give you some design of the studies that we have, I'm going to go through this a little bit slowly so that we actually follow it through as well. So remember I talked about the cross-sectional design of our study. So this would be the cross-sectional section where where participants, we will be recruiting participants. They don't all come in at one time, they'll come in over a period of time, but that is the time point when we are screening individuals. And that time point would be our cross-sectional, our time point would be cross-sectional, And that's the time point where we would be measuring the background HIV incidence. So this would be all our participants that would be adolescent girls and young women who would be eligible for our study. When they come into us for screening, one of the criteria would be HIV, having an HIV test. Once we test them for HIV, all those that would be positive will then have the lag avidity assay undertaken, and they will contribute to the based background counterfactual HIV incidence. These women who test negative then can go into our study, into our follow-up study. They can be randomized to any of these products that we have. However, we need to make each of these products will be tested against a placebo. Now, when we talk about the placebo, we're talking about a TAF placebo and a TDF placebo. So each one gets the intervention. So here they will get the intervention, they will get the lenacapavir, but this group will also receive a TAF placebo and a TDF placebo. So these placebos, the tablet will look exactly like the TAF, the tablet will look exactly like a TDF, but it will be a placebo. Now, in this case, we can provide these placebos because they are getting the active product of lenacapavir. The second group that the woman would be randomized to will be, for example, TAF. And in this case, they will get a lenacapavir placebo. So the active product in this group will be the TAF product. And in the third group will be the TDF, which is the Truvada, and they will be getting the lenacapavir placebo as well. So this is the group that we know will have, we cannot give placebo alone. So we have to give what's existing as part of our effectiveness of our interventions, and as our standard of care. And so the TDF is our standard of care, but they will be getting the lenacapavir placebo. So these individuals will all then be followed up in time. Now this group, the TDF group, because it's our standard of care and the lenacapavir, they're still getting the benefit of the Truvada, and this group will then be considered as our internal active control. So this is our control group. And this group, the baseline, so these women coming in over time will then be contributing to the counterfactual HIV incidence rate, and this will be our external control. So these are individuals in the community who will give us the HIV incidence rate. These women will then all be followed up, and our endpoints will be on two primary endpoints, where we'll then compare lenacapavir to our counterfactual background HIV incidence, and our TAF will then be compared to our counter-based background HIV incidence as well. We will be able to compare to our internal control, but I will show you what actually might happen based on that comparison as well. Sorry, I'm just going to have some water. So if we look at our group, so if this is our background HIV incidence, this is our external control. These are women that came in who were screened for HIV and then had the additional bioassay undertaken on the HIV positive individuals, and we were able, and they had no interventions, etc. whatsoever, and we were able to measure the incidence rate based on the assay measurement. And in this case, our assay showed us that it was HIV incidence of 6.2. So this is indicating to us that we still have a very high community HIV incidence rate as well. Then we would have the lenacapavir with the TAF placebo and the TDF placebo. And for example, at the end of the study, we find that the HIV incidence rate in this group was 1.9. Then we would have the TAF group with the lenacapavir group. And in this case, the incidence rate at the end of the study was 2.2. And in our internal active control group, we had the Truvada, and we have the lenacapavir placebo. And in this group, at the end of the study, after following up these women, with the HIV incidence rate was 2.4. So if we look at these incidence rates, this gives us some indication of the effectiveness of each of these products based on product use as well. But the three would not be significantly different. If we compare these three to the background HIV incidence, that would give us a statistically significant decline in HIV incidence in comparison to the background, and therefore each one of these could actually go forward because we have a statistically significant reduction in HIV incidence in comparison to the background HIV incidence that we have. So can you see what happens if we had to compare, if we excluded background HIV incidence to determine the effectiveness of any one of these would be difficult because we wouldn't have the background HIV incidence rate to compare as well. But what we are hoping to achieve is that over time, it will become even more difficult to undertake HIV prevention trials as we have treatment being scaled up, interventions being highly efficacious interventions being scaled up, and we get community incidents coming down as well. So that's the next challenge that we are likely to face as well. So these are the incidence rates that we have from this group, and this is the incidence rate that we have from the background HIV incidence as well. So this is another sample, and this is a much more simpler sample where we're just comparing the lenacapavir to the Truvada, and again we would have the lenacapavir with the Truvada placebo, with the and the Truvada with the lenacapavir placebo, and this would be our active control, and then we would compare it again, very similar principles, concept. So we can actually test 2 products, we can test 3 products, but to make sure that we have all the interventions and the placebos, again, with our counterfactual HIV incidence rate as well. And this, just to remind you that the counterfactual is a biomarker method and based on a recency assay. So in this case, again, we would have the lenacapavir and the Truvada with a incidence rate of 1.9, and in this case we would have the Truvada with the lenacapavir placebo, and which is our internal active control, which would be around 2.4. Again, very similar. These two would not be statistically significantly different, whereas if we compare it to our background HIV incidence, and if that was 6.2, we would get a significant decline in HIV incidence and would then determine the effectiveness of these interventions as we go forward as well. So just in summary, there is still a need for HIV prevention trials, and it's mainly to expand our choices on HIV prevention interventions and the options that women would have, or anyone for that matter, in terms of expanding their choices as well. Ethically, currently, we cannot use a placebo to test new interventions. And if not placebo controlled, then what? So we have to compare it with some other HIV incidence measurement to the current proven methods as well. And therefore we look, and of course, there are challenges in measuring new HIV incidence rates, specifically new infections. If we use the randomized controlled trial and follow up, it's obviously following up is time consuming, etc. It's costly, etc. But if we have to use any, and therefore we look at opportunities to explore the counterfactual estimates as well. And the counterfactual estimate is your background HIV incidence in the population, in a similar population that would not have had the intervention. And here with the background HIV incidence, we're looking at the recency assay testing in the population as well. So when I looked up, there are several other markers that one can use as a counterfactual, but not for HIV incidence. So for example, we can look at rectal gonorrhea, and if we get a decline in rectal gonorrhea, for example, in the MSM population, and we get a decline in, if we, in the absence of an intervention, we have an increase in rectal gonorrhea, and we have a decline in incidence. So those are some of the comparisons. So I wasn't going to bring in that complexity here, but just to focus on the counterfactual HIV incidence, because currently we still have high rates of HIV incidence in our populations, and therefore we will be looking at the counterfactual. Having said that, I also want to just remind ourselves is that there are several statistical considerations that underpin all these measurements. And I didn't bring that in because I felt that is for the statisticians to guide us. And I will be just losing us in terms of what we actually need to understand as well. And I think that's where I'm going to stop. And thank you very much.
Prof, thank you very much. Thank you so much. There is a need for HIV prevention trials to expand on choices, and we actually have some success, and that success has brought us a number of challenges where ethically we cannot use a placebo to test new interventions. So the question we're asking today is, if not placebo controlled, then what? And that is where Prof has taken us to concepts of comparing HIV incidence measures to proven methods, which are the concepts that we have now. Now, I hope that really has not left any of our heads spinning a little bit. If by any chance they did, do not worry, because we have a number of, we have this forum and we have a number of other forums where we really continue to engage on these concepts, because they are important, and this is where we are at as the field. So it is very important for us who work in communities and who advocate for research, for more choices in HIV prevention, but most importantly, to be able to understand this so that we can engage with the processes of research, trial designs, and understand the ethics that are involved in all of this process. So we hope you really enjoyed this session and picked up some concepts, but if by any chance you have not, we continue to engage. And in fact, one of the platforms that we have presented here is a community stakeholder engagement community of practice. Please take a look at the link that is here and continue to engage on these concepts even at the end of this session. Prof, we have a few questions, and one of them really relates to the concept of placebo. Ngangi is asking, looking at the definition of placebo as a dummy treatment, what happens if placebo is mistakenly administered to participants instead of the drug, or vice versa? And is this something that happens? And if it does, what actually are the ethics of this, and what should we do in such circumstances?
Sorry, are you meaning that, sorry, I didn't hear that very well. So just speak a little loudly for me, please.
Okay, let me try that. So Ngangi's question is looking at the definition of placebo as a dummy. Can you hear me, Prof?
No, you faint.
Am I faint?
Your sound is dying off.
How is this?
Yeah, that's better.
All right. So looking at the definition of placebo as a dummy treatment, what happens if placebo is mistakenly administered to participants instead of the drug, or vice versa?
If it is accidentally, so when we talk about a placebo, it's an inactive product. As it stands, it's an inactive product. So the placebo will be inactive. So there will be nothing happening. What will happen is that should the individual be getting the intervention itself, we might get a drop in the levels of what is actually required for HIV protection. And so by giving them the placebo, the drug levels will decline. And so they will be at risk for at acquiring HIV then.
And
If
how
we're
does
talking about in the context of placebo itself, but we're not talking about placebo in the combinations. Is it in the combinations, or is it? So what basically would happen is that by getting a placebo, you're compromising the level of protection, because you would be decreasing the effectiveness or the availability of drug, of the active drug that would be protecting them. The placebo itself will cause no harm, because the placebo itself is an inactive product. It has no activity whatsoever.
Wonderful. Thank you, Prof. Again, I would like to remind everyone, please look at the link. This is a platform where we continue with these conversations, which is the stakeholder community of practice. It is found, or you can go to engage.avec.org and you will find this. And if we cannot answer any of these, that platform is there for us to continue and really talk about these concepts. Christina is asking, is there harm that can be caused by using placebo as a study participant?
No, there's no harm in the placebo itself. As I said, the placebo is an inactive. It's basically like saline or water or something. It doesn't have an active product. The placebo does not have an active product. But as I indicated, if you use it accidentally in a person who's supposed to be given the intervention, the active product, you're compromising the opportunity to reach the drug levels. But what happens as well is that whoever is dispensing the intervention or the placebo, they, especially at the time of the trials itself, the pharmacy will go through very strict, rigid processes and procedures to make sure that kind of an error doesn't happen.
Does not happen.
Does not happen.
Yes,
it could happen very inadvertently at some point or the other, but there are strict processes and procedures that the correct intervention is given to the correct individual. Whatever an individual has been assigned to, they are provided with that same product, that same intervention throughout the trial.
Thank you very much, Prof. And in fact, Nita agrees that this was an excellent session. And in fact, the messaging of these concepts are needed to explain to community stakeholders and ourselves as members of the research teams. And Elizabeth is asking, and counterfactual trial design has its own limitations, which sometimes an advanced counterfactual analysis would not be enough. How best do you plan to overcome such a limitation in your trial?
I'm not sure what she means by counterfactual. How is it going to be?
The limitations.
What are the limitations?
Liz, would you like Liz to open the mic and ask your question? In the meantime, as we wait.
Okay, thank you.
Yes, Liz.
Okay, Ntando. I meant maybe counterfactual trial as a design, counterfactual trial design as a scientific design, saying that maybe it might have its own limitations as they are conducting their own trial. So I'm saying that how are they prepared to overcome, such a trial, such a limitation if it occurs?
Was that helpful, Prof?
Well, I'm trying to translate it at a practical, at the field level itself. As I said, a lot of this is underpinned by the statistical considerations, and there might be limitations with that, but I'm not going to go into those details. But the counterfactual is, Elizabeth, if you recall, when participants come in, you screen every single individual participant for, eligibility criteria. You would have an HIV test. You will look at the age of the participant, because that's one of your inclusion criteria. You look at the criteria to get into these HIV prevention trials, they have to be HIV negative. So you would be conducting an HIV test on them. Once you've conducted that HIV test, you will be able to identify whether the individual is positive. In that case, that positive individual will contribute to the counterfactual HIV incidence. And if they are negative, they will contribute to the HIV, to the follow-up of the study itself.
All right.
So
Prof,
what I know from my understanding, what I do know where it can impact is that, so there are lots of population-based surveys that have been done throughout Africa. And if many of you might have been involved with them as well, which are the FIAs, the population, the impact assessment, population HIV impact assessment surveys. They've been conducted across Africa, across 12 or 15 different countries, and now they're going through the second round. The problem with the counterfactual background HIV incidence is that you need very large sample sizes to get a reliable estimate. And that's where my concern has been in terms of whether we would be able to get that many individuals to contribute to a robust HIV incidence estimate. Previously, HIV incidence has been based on modeling, etc., but this is going to be on a measurement itself as well.
All right. Prof, thank you very much. And we have really come to the end of our session, but just one more question, and hopefully others can really remain for 1 or 2 minutes. And I don't know if you're able to speak to this, Prof, because Nandi is asking, can you please talk about the study design used in Mosaico study? And in fact, that probably is going to help with really having practical examples in terms of the designs. And
what study?
The Mosaico. And Nandi, would you like to sort of take us through that quickly?
Thank you, Ntando, and thank you, Prof, for a brilliant presentation. So this is the HV10706 study. It's a vaccine trial that's targeting MSM in the US and in the Americas, as well as in Europe.
Thank you, Nandi. And Prof, if you're not familiar with the study, it is fine, but I think it's reason to continue with this, you know, and so.
I must be honest, I know the Mosaico trial, but I haven't looked at the details of the design itself.
Okay. All right. Prof, one parting message for us as community advocates and community outreach workers around these new trial designs. What is it?
Well, I think it's important to know that we won't be able to use placebos on their own. I think we have to continue with our HIV prevention trials. As I said, that we have to expand on the availability of choices. Women should be able to decide, what choice they have and what they would be comfortable with. And for me, I think they are still clearly very, very important because our HIV incidence rates are still unrealistically high. We've been working in the 1990s field, and those incidence rates, those viral suppression rates and the incidence rates are still not coming down they are coming down marginally, but not to the level that where we can get epidemic control in the next couple of years by 2025, 2030.
Thank you.
We still have a long way to go. We always considered this to be our last mile. That last mile is still very, very long.
There's still a long way to go. When I was about to let you go, John wants to ask you a question in person. Over to you, John.
Thank you very much, colleagues. My question really is based on the use of Truvada as part of PrEP. Currently, with the new regimen, we are using TLD, which is a combination of Truvada, lamivudine, and dolutegravir. So I've been asking myself, why specifically using Truvada as PrEP and not lamivudine or dolutegravir?
I think those are used for treatment. Those aren't they used in treatment?
Yes, but Truvada also forms part of TLD with the recent regimen.
Yeah, so the dolutegravir, I mean, I would imagine the dolutegravir, etc., would be highly efficacious, but they haven't gone through the principles of the testing for prevention as well. And I think,
Okay.
you know, and the other concern would be, I think you want to reserve those for treatment in comparison to I mean, dolutegravir, except lamivudine came in recently, but compared to Truvada, which was the existing one. So and the Truvada was at that time tested as part of an RCT and showed the effectiveness. You know, we followed the science, so we actually go through the processes and procedure and determining. I mean, if somebody is bold enough to take you know, the other regimens through the field, I'm sure that could happen as well. I know we were thinking about that at one stage, but we haven't progressed onto that dolutegravir regimen as well.
Okay.
Wonderful. Prof,
Thank you very much, Prof.
Your presentation has really generated a lot of interest on this topic, and I think we will be calling back on you to really talk about this. I think Bulela has got another question, and I think in the interest of time, we really are not able to even go over the 5 minutes that we have taken for this hour. But we really thank you very much. Keep the questions coming, and please take your questions to the Community of Practice Forum so that we are able to pick up and, in fact, bring a lot of topics into this engagement forum, because there's so much that we need to uncover as part of all of the developments that bring a number of dynamics on how we are working when we in search of a choice for HIV prevention. But Prof, thank you so much, and we really appreciate you taking us through this. And in fact, with the demand that we are seeing, we're really going to be calling on you to really talk to us more. Otherwise, everybody, thank you very much. This was the Engagement Forum, the 2nd edition. And in March, on the last Monday of March, which is a month that starts tomorrow, we'll be having another topic. And in fact, please feel free to tell us about the topics that you would really like us to discuss, like you have done today. Otherwise, thank you very much to all of us and all the partners who are making this possible. And thanks to Miliswa, who has provided us this platform to be able to engage and vet RHI on this platform. Otherwise, thank you, everybody. Until next time, goodbye.
Thank you. Thank you.
Bye.
Thank you.
Thank you. Bye.
Okay.